everybody's attention just briefly, please. I'd like to invite everybody to today's Anthropology and Geography seminar series. Today we have Dr. Zach Throckmorton. He got his Ph.D. from University of Wisconsin-Madison, is now the Medical Human Anatomy Director and Associate Professor at Colorado University School of Medicine's new branch here at CSU. He is a paleoanthropologist interested in the evolution of anatomical variation and its functions, and he specializes explicitly in the lower limbs. He has a very robust publication record, including venues like PLOS ONE, the American Journal of Physical Anthropology, eLife, Nature Communications, and the South African Journal of Science. He is a member of the Rising Star team, led by paleoanthropologist Lee Berger. If you're not familiar with them, they're responsible for the newest discovery of our human relative, Homo naledi, which Zach will be talking about today. So please join me in welcoming Zach Throckmorton as he tells us this afternoon how Homo naledi walked.
I was thinking this afternoon, what do I say to be profound and insightful for the first talk I get to give since the beginning of the COVID pandemic. And what I came up with is really glad to be back and be back in person and be able to share our work with folks. Did a lot of this before the pandemic, and it just feels really good to get back into the swing of things, as it were. So thanks for being here. I mean, it's a beautiful Colorado Friday afternoon. It's getting real close to Miller time, and I appreciate you choosing to spend a little bit of your day with me, and hopefully you enjoy this presentation. So without further ado, we go ahead and talk about everybody else who has contributed an enormous amount of their blood, sweat, tears, time, effort, training, expertise to this project. I am but a very, very tiny component of this well-oiled machine. And so oftentimes folks end their talks with acknowledgments, but a lot of what we've been doing with Homo naledi is a little bit different in terms of sharing so much information, making data available via open access. And so I like to, right off the bat, lead with acknowledging these folks in this photo who are the OG cavers and explorers who discovered the remains of Homo naledi. The cave in which they are found, a gentleman like me is not getting very far, so it's very critical that those folks were able to pull those out of the cave, not just for their ability physically to do that, but for all of their training in archaeology and paleoanthropology and cultural anthropology, historic archaeology, wide range of backgrounds of the folks on this team. National Geographic has, of course, helped us out immensely with the almighty dollar. The folks at the University of the Witwatersrand Evolutionary Studies Institute have always taken very great care of us. They've always been wonderful hosts. And we're now up to over 100 people who are contributing to our ongoing work on Homo naledi. So it's an honor to be part of that team and to represent them here today. I hope that I do not bring too much shame upon them. Specifically for the project that I'm going to be talking about, this is my colleague Tara Chapman, who I have never met in person, but we have spent many hours together via Zoom and actually when we started it was via Skype. Now it's via Zoom. She's based in Belgium, so she's 6-7 hours ahead of me, and we always we try to meet real early here and in the afternoon for her so she get home to her family. And I'm an early riser anyway, so it works out. A lot of other people who are co-authors on this paper that's under development, and I really appreciate the invitation from the CSU Anthropology and Geography Department for the opportunity to share this and especially to live stream this. I think one of the things that's come out of this pandemic is the ability to be anywhere in the world virtually is tremendously useful for so many of us in our different lines of work. And honestly, I mean, I get to talk to my friends more frequently as a result of it. The normalization of the virtual happy hour over the last 2 years has been key to getting through this. So let's talk about how people walk. And walking is a fairly complicated process. Usually I'm a big-time wanderer in a classroom, but I've been told the mic is here and the camera is there. I might be able to get a little further away than 6 feet because the way my voice usually projects. And I demonstrate some walking, but you're all fairly familiar with walking because you do it frequently. When we talk about the gait cycle, we divide it into various stages, and at various stages, different parts of your body move differently in order to propel you forward. One of the things that is remarkable about how we walk is the fact that we're constantly falling forward and stopping ourselves from falling forward. And so one way of thinking about human walking is that it's an exercise in continually controlled falling. So I'm going to go through pretty quickly some functional anatomy. That's the basic science part before we get to the good stuff. I teach my medical students, a lot of them always thinking, all right, this is all fine and good, but why are we learning this? And try to save that stuff that's really interesting to them for later, because you got to end on a high note. But these are the 4 joints that are critically important in walking. And from distal, meaning the end of your foot, to proximal, meaning towards your body, you have the big toe joint, and then the ankle joint, and then the knee joint, and then the hip joint. And those are the 4 joints that are critically important for understanding how people actually walk. So to go into a little bit more detail, we can get some actual anatomical drawings here that have some pieces, parts in them, some bones and some muscles with the explanation that takes as long to read as it does actually understanding the diagram, because it's scientific and you don't just look at it and get the point. I always have to atomize these and break them down and understand the very specific pieces, parts before I put them all together. So it's been a fundamental approach to the way that I do my work. Fundamentals, 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 basics, basics, basics. And once you get those down, then you can start putting things together and understanding what's going on. So because the gait cycle is a cycle, and you can start it pretty much anywhere you want, we traditionally just pick heel strike as the arbitrary starting point. You put your foot on the ground, your heel, or your calcaneus is the name of the specific bone that is your heel bone, hits the ground. And when it hits the ground, the weight of your body is being transmitted down through the vertebral column and then the limb that's hitting the ground, and then there's the ground reaction force, because when you push on the ground pushes back on you, which is the extent of what I remember from Physics 101 from the fall of 1999. And so there's important adaptations to the way that we walk that have evolved gradually over time in kind of a piecemeal fashion. We have a decent understanding of the tempo and the timing and what arose when. And so one of the things that's really exciting about Homo naledi is being able to discover and think about in more detail, with more data, how is our understanding of how we developed and evolved our ability to walk, how did that happen? And so when you have this moment of heel strike, you have what is called a flexed hip. In other words, your hip has come up a little bit relative to your body. There's it's one of those ones where it's easier for me to walk around and do this. Flexion of the hip is just bringing the hip up. You have an extended knee. You have an ankle that is in its neutral or just flat bent position, normal position, to a slightly plantar flexed, which is what we call when the toes go down lower than the ankle joint. There's flexion towards the plantar surface. And then you have dorsiflexion of that first metatarsophalangeal or big toe joint. All the other toe joints do this too, but it's much more important to understand how our big toes are working than the other toes for a lot of different reasons. When you get the weight of your body above your foot, that is called the loading response. And at this point, you have a very slightly flexed hip, you have a slightly flexed knee, you have a neutral ankle, and a neutral first metatarsophalangeal joint. This is pretty close to the normal resting standing position. It does not take a whole lot of energy to get into this position or to maintain this position. and then you move forward into mid stance, where you get a neutral hip, neutral knee, neutral ankle, and neutral first metatarsophalangeal joint. This, if you do it on both legs, that's standing. And so the neutrality of all of those joints minimizes the amount of energy you have to expend contracting your muscles, so that's why it's easier to stand around than it is to walk around. When you start to move forward, we call this terminal stance, the end of the stance phase. It's the last part before that leg comes off the ground. At this point, you have an extended hip. The leg is pulled back against the body. The knee flexes a little bit, the ankle plantar flexes, and then the toes also come up. The toes are coming up effectively to prepare them for you to swing your leg forward, because if the toes are falling down, you would trip over your toes, and that would not produce a evolutionary successful strategy. You can imagine that every time a hominin went to go walk forward, they fell over. That lineage would not make it to today. This pre-swing is, again, just getting you ready to swing your leg forward, the maintenance of that neutral hip, getting that knee flexed, getting the ankle plantar flexed, getting the first toe dorsiflexed. This is copy pasta off of my lectures on the gait, which is why there's muscles here. Don't worry, there's not an exam. You don't need to remember what the attachment sites of the second layer of the foot intrinsics are. Finally, you get into the initial and mid swing, where you have a flexed hip, a flexed knee, a dorsiflexed ankle, and a neutral first metatarsophalangeal joint or big toe joint. And then lastly, terminal swing, which is right before your heel hits the ground again. You have a flexed hip, extended knee, dorsiflexed ankle, neutral big toe joint. Sorry, but I wanted to give you a little bit of background on how complicated the interaction of these four joints are through this cycle, because you got to get these information down if you're going to try to understand how Homo naledi actually walked in comparison to how living humans walk. So one of the things that, again, we take it for granted that we are able to walk with a lot of fluency. Walking is really difficult. And I think that it's very remarkable that developmentally, humans tend to develop the ability to walk reasonably well, walk with some fluency, about the same time that we actually start being able to have a human-like conversation. About the same time that you start seeing children develop an ability to empathize with others. You know, kids, when they're 1 year old, if you display pain to them, they don't know what to do. But by the time a kid hits about 4 years of age, if you fall and you injure and scrape your knee because you messed up one of those aspects of the gait cycle, you know, a kid will come up to you and try to comfort you, because that's an innate character of humans, is to cooperate and take care of each other. So this is my nephew, Carson. He's going to be turning 4 this year. And it's been really astonishing watching him develop from, you know, an infant or a newborn that is, well, there it is. We got to take care of it. And my poor sister and brother-in-law, they have to do all the hard stuff. I just get to see the smiling photos for the brief moment he's happy at the pumpkin patch, and not the hour he was screaming that he wanted more ice cream. And just like being able to talk with syntax, grammar, being able to communicate our feelings to each other in a way that is different than other animals, walking defines us as humans, and arguably that's one of the important definitions of what makes a hominin a hominin. In my opinion, and I have argued, you know, if you're not walking bipedally, you're not a hominin. And so there's lots of arguments about the stem hominins, the earliest hominins. Are they a hominin or not, based on what the limited amount of information we have about how they walked. But fortunately with Homo naledi, we got a lot of information, and we have a good idea about how they walked. You know, walking is so fundamental that it gets to the point where you can identify the walking cycle, the gait cycle of people you're close to. If you worked on the office from somebody for years and years and years, you know who's walking down the hall based on their footfall, based on the tempo of their way they walk. Even when you go away for 2 years because of COVID and then come back, you know who people were walking around. And like many other fundamental aspects of who we are, when we exaggerate it, it becomes a source of humor. And so my students over the past few years started busting my chops about my outdated pop culture references. And so I reached into the Wayback Machine. This predates me, but it is classic. I tell them it's not outdated, it's classic. This classic sketch from the second season of Monty Python's Flying Circus, where he's greatly exaggerating multiple components of the gait cycle. And that was, in fact, that used to be a question I would give my medical students is, watch this video and then tell me where are the exaggerations of different parts of the gait cycle. And I'll just be like, you gotta be kidding me. Then they'd look and that's actually pretty easy. There's some really obvious hyperflexion of the hip going on right there. So over the next 14 slides, I want to review all the neuroanatomical considerations of that. Just kidding. A little more updated pop culture reference. This came out when I was in high school. We're not going to do that neuroanatomy. If you're interested in that, you can take BMS 345 and we can chat about it, or you can go to the CU School of Medicine and we can chat about it. So let's talk about the fossils. Homo naledi means homo or same or human. It's in our genus, and naledi is from the Sutu, which is a local African language word for star. So again, with the outdated pop culture references, when they share what we're going to call this thing, I immediately think of the old Jeff Bridges movie Starman. It's like, don't say this out loud. Just don't say it out loud in the room with everybody else. I did bring some 3D prints of the articulated foot and the articulated hand, so we can pass these around and you can get an idea. These are 3D laser scans and then 3D prints. That is very close to what you work with when you actually handle these specimens. Homo naledi was discovered in the Cradle of Humankind, which is the site of many important fossil discoveries from coming up over 100 years ago now. Do I need to remember these dates? Well, yes, for this exam, but then you'll probably forget them like I have. About 100 years ago, all the way up to much more recently, there are more than 1,500 fossils that we can identify exactly where they came from in the body, and there's still a whole bag of small fragments that we haven't been able to identify and are probably of limited utility. The specimens are continually recovered. There was a pause, of course, for COVID, but they are resuming the excavations, and so there are continual fossils coming out of the ground. It took a few years because archaeology can be exceedingly difficult sometimes to figure out that Homo naledi was somewhere between like 230 and 330,000 years old, which is like pretty remarkable that something that has so many old features anatomically would be presenting as recently as 250,000 years ago. But with the discovery of, say, like Homo floresiensis and Homo naledi, I would put money on the fact that we're going to continue to find unusual, ancient-looking hominins that probably persisted to fairly recent time periods. And there's going to be folks who are going to be finding these through the next couple of decades, and I think it'll be thrilling when they do. But bringing it back real quick, for the purposes of how we figure out how Homo naledi walked, we have multiple first pedal proximal phalanges articulating with first metatarsals. And so we have multiple first metatarsal phalangeal joints. We have multiple ankles that include the talus, the calcaneus, the tibia, the fibula. We got everything down to the malleoli, the big bumps on the sides of your ankles that are important in stabilizing it and are important sites of ligament attachments. There's great knees. There's multiple great knees. We got great patellas or kneecaps, the big sesamoid bone that develops in your quad tendon that is critically important for you to be able to walk. When you extend your knee, get that force transferred from your leg, from your thigh to your leg. Hips are a bit of a different story. there are not great hips, and so that's been something that has limited our ability to recreate how Homo naledi walked. There have been some hips that have been discovered since the initial findings that have been helpful and instructive, but without belaboring the point, it's not surprising that hip joints are not surviving particularly well because of the specific bony anatomy of the hip joint. These specimens were all discovered in 2013, so we're coming up on the 10-year anniversary of the discovery of Homo naledi. It was published in 2015, and it was just completely overwhelming. We talk a lot in academia and professional work these days about imposter syndrome. Do I really deserve to be here? And one of the things that helped me with that was just recognizing that I'm just a small part of a big team, and everybody deserves to be here, and everybody is contributing. And when you have that many people working that hard for so long, you get pretty spectacular results. So let's talk about where these fossils were found, and then we'll talk about the anatomy, and then I'll show you some of the most recent work we've done. The Cradle of Humankind is in the northeastern part of South Africa, which is in southern Africa. It is right outside of the major city of Johannesburg. Johannesburg is a bustling metropolis. It is a fascinating place. You have so many different cultures from all over Africa, all over the world, mixing there. You can roll over to one place and get some really great Italian food. You go across the street, and now you can get burritos. When I first got there in 2014, you could not. But importantly, now you can get burritos across from the Italian place with all the other traditional African cuisines. And my students tease me, they're like, all of your histology teaching is comparing these slides of cells to food. And it's like, all you talk about, and I'm like, yes, I'm a very food-motivated person. This cradle, it really blows my mind that the Cradle of Humankind, where you had these very similar yet very different hominins living for literally millions of years, right down the road from Sandton, which has got the best mall I've ever been to. And they have this, two-story, Cinemax or whatever you call it, Dolby surround theater, and there's just everything is so 21st century, hyper modern. And you get in a car and you drive 50 minutes to the Cradle of Humankind World Heritage Site, which is called Maropeng, and you could see this work being done on our ancient ancestors that if you brought them forward in time, I don't know how they would react, but I think it would probably be astonishing to them. They'd probably be going to the food court first, just like me. When you visit the Cradle of Humankind, they have so many different specimens available for study. And I like to highlight my friend Kim, who is curator at Maropeng. She's also interested in lower limb biomechanics and anatomy, and she's been working as curator at Maropeng for about a year now. And I hope I get to go back next year and see my friends there. The specific work that we've done has taken place mostly at the University of the Witwatersrand, which is in Johannesburg. That is their big, impressive main building, which looks like it dates to about the same time period as Clark. With the fountain, there's the evolutionary study. Actually, the Evolutionary Studies Institute, that looks more like about the same time period as Clark. When I was an undergraduate student, our anthropology department was in the building that was built in the late 60s and had that same circular cement stuff up front that I still like. Is that for large bees or something? I don't know what that's there for. But that's the Evolutionary Studies Institute at the University of the Witwatersrand. Down in the lower left is my buddy Byrne, and in the upper right is my buddy Cephalani. Cephalani is collections manager and Byrne is curator of the ESI. And they always take such good care of us when we come down there to work with the fossils, and communicating with them is always, just straightforward. And I'm looking forward to hopefully seeing them again next year. When you go into the Evolutionary Studies Institute, the fossils are stored in a vault that is protected by a bank door. The picture above the vault door is Philip Tobias, who is a legend in paleoanthropology. He's a legend in South Africa as a champion of civil rights, the rights of black South Africans and native South Africans. He passed away somewhat recently. And once you get into the vault, there's all sorts of specimens. The glass cases, that's one of the Australopithecus sediba specimens. The small head in the glass case in the center is the original Tong child. So being able to actually see these real specimens in real life, I've actually never touched them. That would be like, it's like, it just feels strange, like they're such hallowed relics that someone like me, not even with washing hands and gloves or anything. And then you could see just drawer with all sorts of useful and well-known and well-studied hominin fossils.
Like John Hawkes?
Like John Hawkes, yeah. That's my doctoral advisor from Wisconsin. We were all younger when I started working there in 2007 with him. Fossils like John. So this is just a screenshot or a couple of photos of when we were actively studying these fossils. These are a bunch of folks who are chatting with my friend Kim, who is in the left on there with a kind of look on her face. Pretty typical of Kim. I don't know if she's watching. We know each other well enough. I can get away with that. That's her in the upper right-hand corner. We spent lots and lots of time putting together these fossils into feet that we had reason to believe from both the taphonomy, where they recovered with the records from the recovery team, the archaeologists down in the cave, and then confirming that they were anatomical good fits for each other. You can see our extraordinarily high-tech Tupperware containers and bags of rice from the local supermarket, but we do that because it works. So let's talk about these joints. This is a femoral shaft. This is a thigh bone shaft on the bottom. That's the proximal part of the femur, the thigh bone on the top. There's a good femoral neck there, but there's not much of a femoral head left anymore. Mostly because the cortical bone on the femoral head is fairly thin, and once that's gone, the spongy bone underneath just erodes. That thinner cortical bone on top of that heavier spongy bone is an adaptation for weight bearing, gives the joint a little bit of spring so that it's not super rigid and gets broken apart by high forces that are going through it. And we really only have some of these scrappy bits of the pelvis, the os coxa, the innominate bones, the socket of the ball and socket joint of the hip. But we do have some decent acetabular sockets. There's nothing from the scrappy femoral heads and the okay acetabular sockets that indicates that Homo naledi's range of motion, how it could move its hip joint, was fundamentally that different from ours or pretty much any other hominin. The knee is superhuman looking. That has we call this a valgus angle. This is an angle that helps to bring the feet narrowly underneath the body. So our hips are typically wider than our feet when we stand. That is something that biomechanically and bioenergetically helps us conserve energy as we walk. You could test this by just walking for any period of time with your feet spaced about a foot and a half apart, and you're going to notice that that's going to tire you out faster than putting your feet together. That is a kneecap, that's a patella. It's very thick from forward to backward. The anteroposterior dorsal ventral depth of this patella is very human-like. There's a big lever arm, big joint cushion for the knee. And we have multiple patellae, including some from some kids, and they all look very human-like. This is a tibia or shin bone at the top and a fibula, or the other bone in your leg, on the bottom. Fibula is really only important in stabilization of the knee and in the ankle. If you crack that thing and it's not too messed up in the middle, they don't even cast it. As long as the knee and the ankle are still in good shape. This is a shot of a medial or proximal medial aspect of a tibia, so like right here. This is a site of attachment for an important anatomical structure called the pes anserinus, or goose's foot. And at the goose's foot, you have three muscles that come together and share a common tendon that then sticks on this part of the leg. That's awkward. It's like the first time I've tried to do this thinking, okay, is this going to work? Yes, it worked reasonably well. And each of those muscles is important in stabilizing the leg. And in Homo naledi, it's a raised bump. That's weird. That's one of the weird things about Homo naledi's lower limb. Like was mentioned, my particular interest is understanding the breadth of human anatomical variation. How do we differ from one another, from our muscles to our vessels to our bones? And I tell you, I've looked at hundreds of human tibiae, but that'd be a lie. I've looked at thousands of human tibiae, and I have never seen a living human who's got a bump right here. And so I think that it's very interesting that the site of attachment for this three-headed muscle group is a bump on Homo naledi and on nothing else, not any living human and nothing else in the fossil record. I had no idea what this meant initially. I think, given some of our more recent analyses, I have an idea of what's going on here. And then this, of course, is my favorite part, the foot. And so this is foot one, the specimen that's being passed around. The only thing that's missing out of this foot is the medial cuneiform. But in fact, we even have the little tiny sesamoid bone that goes underneath the toe. It is the only flexor hallucis brevis sesamoid in the hominin fossil record. I found it in a bag of sediment from this. There are 3 other people in the world who care. But I was like, hey, I got the only one. It's completely inconsequential and meaningless bone, but I contributed something. The transverse tarsal joint, or the joint that connects the ankle part from the midfoot, is very human-like. It has multiple bony mechanisms that really lock it down when the creature it's a hominin, when the guy or the gal or whatever put its foot down. There's a transverse arch from side to side, which is very hominin-like. Is important discoveries that were made during grad school about the Australopithecines, that even the ancient Australopiths, 3 million years ago, had a transverse arch on their foot, so it's arched not just from front to back, but from side to side, which is an important loading mechanism, or mechanism for being able to accommodate loading during walking. And we have reason to believe that there was a longitudinal arch from front to back, so the typical arch that you think of when you think of the arch of your foot. I think that they probably had a little bit of an arch. My friend Will thinks that they're probably closer to like a normal or healthy flat foot. We respectfully disagree on this point. I mean, if we find more fossils, maybe we'll be getting some answers one way or the other. But honestly, I don't think it really matters, because fair amounts of living people have a normal, healthy, flat foot, and lots of people have normal, healthy, minimal arches. So, again, it's not just that specific aspects of the anatomy match living people, it's that ranges of variation match living people too, which is so weird, because when you look at the head, you'd never mistake that head for a living person's head. But I will tell you, when I first got photos of these specimens, my heart was bumping and my stomach was churning, because I thought that these were kids. From the photos I had been sent, the feet bones were so human-like that I was like, this is some sort of, I don't know, P.T. Barnum nonsense. But then I saw pictures of the head and I was like, no, this is definitely not human. This is not a kid. So to have the feet be very, very human-like and have other parts of the anatomy not be human-like at all. But then when we started studying aspects of the foot, some interesting things started arising. And so we have many, many foot bones. This is what was from the initial publications in 2015. We have additional specimens now that importantly are very consistent with this. We haven't found anything that's like, wow, this heel bone is really different from all the others. So everything that we found, at least from the lower limbs, has been pretty consistent with the initial stuff. The calcaneus, or the heel bone of Homo naledi, looks very much like Australopithecus sediba. It looks very much like Australopithecus afarensis. It is getting towards the level of robusticity displayed by living people. This character is, we're pretty certain, mostly a function of the fact that, you know, we got bigger and heavier as we got more and more towards the present era. And the reason why we have bigger calcanei as living people than, say, naledi, sediba, or afarensis, because we're bigger than they are. And that's very different from gorillas and chimpanzees because they are not walking bipedally. They don't need this big, robust calcaneus to adapt to the ground and to accommodate those ground reaction forces when they walk. I mean, gorillas, with an N of 20, we got 400 or 500 pound male gorillas in that sample whose heels are smaller than small human heels because they don't walk like us. The calcaneocuboid joint is the specific joint in the lateral aspect of the midfoot, so on the outside of your foot. It's got this beak on it on the inside and on the bottom. It's a mechanism that locks the midfoot and provides stability so that when you go forward over your foot as you go through the gait cycle, your foot stays rigid and does not start twisting on itself in the middle. I'm working on a paper with some folks over in Europe looking at how the ligaments in these joints contribute to stability, and this has been a particularly interesting one because our experiments on anatomical donors or cadavers highly indicative that you can cut like both the dorsal and medial calcaneocuboid ligaments and then the ligaments that attach this joint to the talus and navicular, and it doesn't matter because the bones are what is keeping the range of motion from moving too much. So again, this all indicates that there's reduced flexibility in the foot and that there's more stability, especially during heel off as you move towards putting your weight right over your foot. The hallux, or the big toe in Homo naledi, is adducted. I always put that second as a capital because adducted is the opposite of abducted, and it's one of those things that even I get tripped up. Basically, all this is saying is that unlike this chimpanzee foot in which the big toe is abducted or off to the side of the long axis of the foot, in Homo naledi, the big toe is brought in line with the rest of the foot, just like it is in your foot and the foot of living people. All of the medial cuneiforms, which is the important joint for determining this, they all look very human-like except they're small, and they all indicate that naledi had an adducted hallux. It also had a long toe. The big toe is fairly long, just like it is in humans, and you can see naledi is clustering right by school 4, which is a Neanderthal. I haven't taught paleoanthropology in 2 years. Which site is which species? And then you can see LB1, which is Floresiensis, is weird and unusual out there, but naledi is very similar to Neanderthals and humans with that relatively long big toe. But while its toes in its medial column, so the big toe, the hallux, the first toe, and the second toe were very human-like in that they're different than the third, fourth, and fifth toes, which appear to be too long. So a few years ago, we published a study using statistics I don't understand, but were approved by multiple statisticians on the paper and our peer reviewers, so I believe in them, showing that while the relative length of the hallux and the second toe compared to the first metatarsal and the second metatarsal were within the human range, the third, fourth, and fifth, or the outside digits of the foot, those toes were too long. So they had these lateral toes, or these minimal digit, or third, fourth, fifth toes that are a little bit longer than humans, but they're a lot shorter than chimpanzees. And so I think that this is particularly interesting because it shows us that it looks like the medial column of the foot probably evolved to have shorter lengths and be more like a human before the outside of the foot did. And one of the things that I also think is interesting is that of all the specimens of naledi that we have, there's a fairly high frequency of broken small toes. And you could see the breaks on the phalanges. And at the time, we didn't know this, but looking back on it now, I gotta wonder, is one of the primary reasons why we have short toes off to the sides so they don't stub our feet. Like, okay, well that seems like a pretty mundane reason for how our bodies came to be the way they are, but maybe that is in fact what happened. So this is work that I did with Sarah Trainer, who is now a Ph.D. and teaches anatomy at the University of Wisconsin Medical School. She and I overlapped for 2 years at the University of Wisconsin, and she did her undergrad here. So she is an alumna of the CSU Anthropology department. And I kind of wondered why she stayed in Madison after she finished her Ph.D. And then I came here. And I realized that if you did your undergrad in Fort Collins and your PhD in Madison, Wisconsin, you ain't ever leaving. So these are just two of the greatest college towns in the country to be living at. And winters here are a little bit easier because the snow piles melt away in 2 weeks, as opposed to Wisconsin where it's 0 degrees and the snow piles don't melt, even though it's sunny. The specific mechanism for how the toes bent in relation to the foot, very similar to humans. You can see this extraordinarily high-tech method of determining the angulation of a toe base. So this was one of those ones where you train on sophisticated methods of analyzing specimens, and then you actually get there, and the 3D scanner won't scan this bone because it's too small. And so no matter how you try to attach it to the plate that's turning it so that the 3D scanner can scan it, you just keep getting all these artifacts, and it just doesn't work. And after like an hour of this, I'm like, well, how did they used to do this? So I looked up, and they got a protractor. And I was like, shit, that'll work. So we set up this device where we just we anchored the camera and we just put it right under it, and I used the centering point by dropping a piece of string from the camera. Gravity will take care of that for me. Like a plumb bobber. Is that what a plumb bob is? Yeah, I don't know where that knowledge just came out of, but remembering, it was my archaeology training. That's how I know what it is. And then taking a pin, laying it against the back of the toe where it articulates with the foot, and then just taking a picture and using free imaging software to calculate that angle out. And I was like, well, hey, this will work. Some of my colleagues are like, you did what? It's like, hey, can't argue with results. So let's talk about how we've been putting Naledi's gait together. So the first part of this is putting together a lower extremity from many pieces, because while we have a really good foot that we're confident from both the archaeology records of where it came from, we don't necessarily have an entire lower limb that was found in situ altogether. Otherwise, this paper would have been published years ago instead of continuing to go on. And so the way that we've been able to do this so far is by putting together feet and long bones. And unfortunately for some of these, some of the best specimens are actually juveniles, so then we have to reconstruct what the juvenile would be if we're scaled up to an adult. That's a whole different set of issues. That's what my buddy Chris Walker does. Yes, his name is Chris Walker, and he helps us study walking. It's almost like my colleague, there's an anatomist whose last name is Organ. That's like, all right, well, I mean, your fate was sealed upon birth. So this is a leg, or shin, and then the foot, just showing you the different pieces, parts that were put together. The second thing that we do once we have that is we reconstruct the ranges of specific motions at specific joints at specific times in the gait cycle. So this is a fundamental understanding of how did the joint move, and then how is it moving at specific times in the the gait cycle. And this is the ankle. So compared to 3 human males, a human female, and 2 human juveniles, it is very difficult to distinguish how Naledi's ankle compared to a living human's throughout the entirety of the gait cycle for the important consideration of the ankle going up and down, plantar flexing and dorsiflexing, the ankle going in and out, so how it turned its heels or turned its ankle in and out in adapting to an uneven substrate. You know, when you're walking on flat ground, your ankle doesn't do a whole lot of tilting in and out. But when you start hiking or walking on uneven surfaces, you got to start turning your foot in and out to be able to accommodate that. And then also the way that it rotated either internally or externally, which is getting into the weeds for this talk. It's so human-like. It is effectively within the range of human variation for the entirety of the gait cycle at the ankle. And I thought that that was fascinating because we had argued in the original 2015 paper, this foot is super human-like and this ankle is super human-like. And we were right. I mean, this is just a very clear demonstration of how human-like this joint actually worked. And then, when you start feeling really confident about what you know, you get results that you go, ugh. So this is the knee compared to the same 3 human males, the human female, and the 2 human juveniles. And Homo naledi is similar, that is within the range of variation for the amount of the knee flexion and extension. So flexion and extension of the knee is just knee goes up, that's extension, knee goes down, that's knee flexion. And I do that in my class. My students make fun of the fact that I can't even extend my leg out towards horizontal. And I'm like, I'm sorry, I don't do yoga. Like, I sit at my desk. That's where I do my work. But they tended to adduct their knees more during the swing phase. And so when they swung their feet forward, they brought their knees closer in towards the midline than we do. And that's odd, because you could see that there's a fair amount of variation between people on how much their knee goes in towards the midline or out away towards the midline during the swing phase. We thought that this was probably just a function of the fact that Naledi was short, but then you compare it to short kids, and it's not like them. And so that's odd. But I think that it helps us understand, when I talked about that goofy bump on the inside of the tibia, the pes anserinus, those muscles are in a perfect place to limit that, or to accomplish that larger degree of adduction. And so I have to wonder that when Naledi put its leg forward, that there was some sort of torsional force or some sort of other force maybe generated at the hip that put their knees out. So they developed very strong medial muscles on their thigh to pull their knee back in. I don't know. 2 of the reviewers on the paper thought that that was an interesting idea, and one of them said it was nonsense. So we'll see when we go for another submission. And then Homo naledi shows more external rotation on the knee joint during the gait cycle. This, again, is pretty variable in people. The amount of external rotation that's done at the knee is actually not that much, and so small differences can make major percentage change. I still don't know what this means. We're kind of just punting that, saying that it's not, that it matches kind of the same magnitude and direction of some of the living humans. I can't remember if somebody had come up with that and put it in the paper. I don't know. I've been working my butt off teaching the medical students, so I actually have only gone through the draft that came out about 3 weeks ago, instead of the one that I got yesterday, that I told them, I'm finishing my class, I'm going on vacation, and I'll read this later. So this is the human reference model that we have for our animation. So this is a 3D kinematic model that was generated by Tara Chapman, who you saw on the 3rd slide of this presentation. She is the primary worker on the development of these models. She's done this before with Neanderthals with really good success. Those papers have all been published and well received. And so we've been trying to now apply it to naledi. So again, this is just a human walking, showing in detail how these joints are moving at different parts of the gait cycle. And then this is how Homo naledi walked. And it's amazing that it can walk so well considering it has no femoral heads. But if you'll bear with that, it takes a very keenly trained eye to pick up that the knees are acting a little bit more differently in naledi than they are in living people. But if you watch the knees, they're going to go outside a little bit more than what you saw in the human. The ankle is pretty much the same in the first metatarsophalangeal joint. The toe joint down in the foot is the same. And I see Professor Dew squinting. You and I could chat and I could show you this and we'll magnify it up so that you don't have to squint as much.
I don't know anything about morphology.
Okay.
So, yeah.
Or you could just accept everything I'm telling you at face value.
I will do that.
So we have another view of this gait cycle. Again, just demonstrating the very human-like ankle, the very human-like toe, kind of unusual knee. And of course, one of the major limitations of all of this work is that we don't have a great hip joint. And so most of the criticism that we have received on the earlier drafts of this paper has been like, you don't have a great hip. And we maintain that there's still utility here because you can understand these other joints in and of themselves. We scaled back some of the claims, as is often requested by reviewers when you don't have all the data that you need or that they think you need to be able to make them. But one of the things I wanted to emphasize is that if you are thinking about going into a career, for the students in the audience, if you're thinking about going into a career in this, you gotta understand this is a long process. This could be a long slog. And I thought to myself at the time, man, I've spent 1.5 years on my dissertation. Well, that was 2015 and here we are in 2022 and the work is not yet complete. The paper that I have been sharing information from has 17 authors from 7 countries on 3 continents. And so in a way, COVID has actually helped us become better communicators across these time zones. But there have been other challenges. Almost everybody on this team's been sick or their family's been sick. And then you lose 2 months because the part that you need to get done can't get done. And it's because they're sick. And because I'm not a monster, I mean, you don't just keep sending emails. My medical student in the front row is like, okay, well, I've yet to see that. But, you know, a couple of us have changed jobs. I came to Colorado State University 1 year ago. 2 of the folks have finished their PhDs. 4 of us have been promoted from assistant to associate professor. A bunch of people have had kids. And this paper was rejected by a splashy journal last year for not being adequately high profile. That was the exact word. Inadequately high profile. And I'm like, all right. After 2 rounds of reviewers' comments, so I was like, thanks. Can you tell us, did the reviewers want to tell us who they were? Because 2 of them we'd like to transfer to the next journal. That one, no. And we're submitting this to a different journal very soon, but I'm at the point in my career now where my vacation, my road trip down to Santa Fe to see the original Meow Wolf and eat some enchiladas and drink some beer and go to Great Sand Dune National Park is now a higher priority than making sure this goes out the door immediately. But if you find yourself in the Dallas-Fort Worth area anytime soon, there is a new exhibit called Origins. It is, as it says, fossils from the cradle of humankind. This is one of the rare times when fossils from Africa have been brought to the United States. Lucy made a grand tour a number of years ago. Lucy is not part of this exhibit. This is all stuff that's from South Africa. It's at the Perot Museum of Nature and Science in Dallas, and this opened a few months BCE, or before the COVID event. And at the time I was in Arkansas, so my wife and I jumped down there real quick, and we got to see this exhibit. It was super cool, all these 3D prints of various and assorted hominin skulls. Neo, who is a partial skeleton of Homo naledi, is on display. The radius and the ulnar are in the wrong spots, but other than that, it's nicely done. The display shows the kinds of animals and plants that lived alongside Homo naledi in South Africa about 250,000 years ago. There's a 3D printed model of the cave system with some of the gear that was used during the original excavations, for safety purposes and all that jazz. There are a bunch of displays, and they were showing a video which included our gate animation. So that was really nice to see all of that in Dallas, again, before hit the fan with COVID. And hopefully, as we continue to move forward, we're going to be able to share more and more information about Homo naledi. This is the most of the original Rising Star group that worked for many, many hours in this vault and have continued to work for now many, many years. And if you are a student, we have many, many, many more years worth of work with this material that is yet to be done. I am currently working with a couple of junior colleagues now. It still sounds weird to say that. A couple of junior colleagues on incorporating naledi data into their dissertations and their early career research. So by all means, if you've got ideas that you want to chat about this, you just let me know and let's chat. I met Tara, who is the major author on this paper, because she emailed me and said, I would like to work on this. Let's chat, and here we are. So thank you very much for your interest, and that's that. Sorry, Mike, I gotta say, working in Joburg really helped me prep for coming out here, because Joburg is high and dry. And the thing you learn working in South Africa is you don't ever go anywhere without 64 ounces of water. Well, I'm fairly familiar with campus here, and I figured I could get away with only 16 ounces. But man, it's difficult being high and dry sometimes.
So I'm sure we have time for any questions you might have.
I was just wondering if, given what you know about the knee, would you anticipate differences in the hip joint? I mean, could you predict some differences in the hip joint if you eventually find them?
And boy, I would love for them to send me a scan of a hip that would confirm the part of the manuscript that is reserved for the confirmation of what the hip looks like. So yeah, we have some ideas. I hope that when we actually do get a really good hip that it'll confirm a lot of what our current thinking is. But I'm not really optimistic that we're going to get a hip that's in good enough condition for us to be that specific. Or of course, what's really going to happen. I got all my predictions. We're going to find a hip, and the predictions are all wrong. Which would also be fun. That's a master's thesis for a young student.
So I'm wondering, to me, the reason the gate looks like this would be because they preserved some wide-bodiedness that's more in that archaic category of features. Does that make sense to you, that you have more rotation at the hip, and so there's just more movement in the upper leg?
They're trying to get around the problem of having a narrow support base and having their feet close together by themselves while having a wide body and a wide pelvis. And we do that in part by getting taller.
So has anyone looked at Australopithecine gait because that would be your best?
And in fact, Tara has started actually putting together that next project with trying to reconstruct an afarensis. The problem is that the Australopithecines have marked body size differences that don't really present themselves in the same way in naledi, and it's tough to put this. And it's tough to put it. It's tough to put together a reasonably complete lower limb with australopithecines. There is not as much material there, and this is exactly why Tara was super interested in this, because, like I said, her first work on creating the reconstructions of gaits and hominins is from Neanderthals, because we got a ton of Neanderthals, and so we have the pieces parts to be able to put a lower limb together. But yes, I think that a lot of this has to do with the mediolaterally wide, ancient body plan.
Right, okay, good.
How fast could naledi run?
Probably not nearly as fast as a living human. And by probably, I mean definitely. We have no indications that one of the major determinants of the speed of any animal, the top speed of any animal, and especially a biped, is how long are your legs. And they were short, and that would have inherently limited their top speeds. There are certain adaptations in the human skeleton that facilitate endurance running. There are other adaptations in the human skeleton that I think pretty clearly facilitate a pretty quick burst speed, being able to take off and run real quick. But the bottom line is that if you're a human and you're running from predators, you're already done, because they're going to get you. So the top speed, we have not reconstructed that. And you know what? I think that would be really interesting to do that. I'm going to give you my card. You're going in the acknowledgments if we get that figured out. I had never even thought, but that's really interesting.
Yeah, that's a good
and we should be able to do that. Yeah. I'm thinking of that scene in Jurassic Park with the T-Rex chasing the Jeep. I don't think Homo naledi ain't running down a Jeep. That I can tell you that. It probably would not be that different from a human child at about the same height. But there are limitations on kids' strength due to their musculoskeletal system that an adult doesn't have. Like Osgood-Schlatter disease, Martin.
How likely is it that the bump on the tibia is due more to genetic drift rather than an adaptation?
It's a great question. I wish I could give you an idea about the inbreeding coefficients and the magnitude to which genetic drift might have been altering specific components of these hominins' bodies. But I read that paper when it came out 3 or 4 years ago, and I cannot remember the least bit of detail on how related they might have been. We have not been able to do that with DNA. We've been able to do that with morphology, but I don't remember off the top of my head what that was. If you looked up naledi inbreeding or naledi morphology inbreeding, I think that the paper should come up as the first indicator. I mean, any one of these particular traits could be the result of genetic drift rather than selection. And my entire dissertation, I went into thinking I'm going to find signatures of selection, and what I in fact found was tons of signatures of no selection at all. And I think that, and in fact, a lot of my research program, starting from my first paper, which predated my dissertation, was why is it that there are so many parts of the anatomy of human feet that look the same across wide spaces of land and across wide distances of time compared to things like heads? You would never in a million years confuse a naledi from a Flores from a Neanderthal head, but you can confuse Neanderthal feet from our feet. And you can even confuse a naledi foot for a juvenile human foot. And so that's been a major part of my, I joke I went to grad school to find selection and all I found was drift. And to this day, that's really all I see it's not all, it's a lot of what I see in the anatomy lab too, when I work on the anatomy of people who are very recently alive.
Yeah, in your sample of, I'll take this down, in your sample of human feet using modern humans, how many of those were people who grew up without having shoes on, or?
That is a great question. And in fact, there have been a series of papers that have come out over the last decade. Very, very fortunately for me and what I have done through my career, demonstrating the bony anatomy between people who are shod versus unshod, very similar. There are important differences in the function of joints and ranges of motion at joints that are the result of probably laxity of ligaments in people who do versus do not wear shoes. But when we look at the patterns in those, it's basically just taking the range that exists in the people who have shod feet and bumping it out a little bit. And so if we included down to the level of the detail of these analyses and the 3D kinematic reconstructions, Naledi would still be comfortably within the range, and arguably even more comfortably within the range, because like I said, the laxity on the ligaments is not changing the underlying bony architecture. But that is a great question, and yes, there have been a few papers that have come out over the last decade where I read the title and my heart skipped a beat, and I read the abstract and again I went, because if you can imagine, if the bony anatomy of people who wear shoes is fundamentally different from the bony anatomy of people who don't wear shoes, like pretty much everything I've done in my entire career is rendered moot. So yeah, I was very happy about that.
I'm just thinking about growth and development and the development of the lateral portion of the foot.
Yeah, and that's difficult to ascertain because that involves the detailed study of juveniles and kids, and as you know, it's really hard to study living kids. So most of that, almost actually, as far as I know, all of that research is focused on adults and not on children.
Are there any other preserved hip specimens, say like Homo floresiensis, right? I know quite a small hominid, but that's been able to kind of inform your decisions about what the hip might have looked like, if there's any similarities there, or is it completely different morphology?
I will be perfectly candid with you, and I will tell you I have not really thought about hips in about 2 to 3 years now. There are probably people in this room who know the answer. Micah, you?
What about floresiensis?
Yeah, I don't.
So I think if you're talking about a biped, you're fundamentally in the same. You're not going to get a lot of the variation in the pelvis is in the upper part of the pelvis. It's how the blade flares out or comes in or moves around or in front. But the joint itself, maybe there are slight differences in depth, width of the pelvis, width of the bidascial plane, but bipeds are bipeds. You don't see a ton of difference.
And one of the fundamental recognitions of functional anatomy is the idea that a joint is inherently a trade-off between flexibility and stability. And if a joint is stable, it's not flexible. And if you're going to be a biped, you can't have a flexible pelvis. Right. You just can't. It won't work. You're going to have catastrophic problems. And Mike, I don't know if you remember this from some of the papers on the thorax, but the attachment sites for quadratus lumborum on Naledi are all really well developed. And I have to wonder, are they using that to act as brakes on the rotation of their hips on their vertebral column, because they have wide hips and they're trying to maintain that torsion all throughout the entire lower limb.
To me, like, this is the one thing I want to say, and then I'll shut up. But if you want to start thinking again about selection, when we teach about bipedality, all of us do the same thing. We always use chimps as the out group.
And we should be using gibbons.
Well, when you force them up on 2 legs, why are they not great at bipedality? Well, because they have a lot of hip rotation. They don't, they're flexible, but they also expend way too much calories in that stance. So if this is a wide-bodied hominin and they have a lot of rotation at the hip and their knees are moving like this, that spells to me a clear selection story.
And it's energetics, and it's a lot easier, it's a lot less costly if you can limit motion with bone. It's a lot safer if you can do it with bone than you do it with the ligaments, because bones can heal, ligaments are not very good at that. And so I think that is one of those areas where, honestly, I think that a lot of the evolution of the hominin lower limb is as much due to joint safety as it is energetics. A human that can run 5 versus 6 kilometers an hour doesn't matter. A hominin that has a blown knee is toast. And if you have stable joints, you are much less likely to have a catastrophic injury to any of them. And then one of my favorite sayings is, you don't have to be the fastest person to survive, you just don't want to be the slowest. And that's why they invite me to these things.
So let's thank Dr. Throckmorton. And